Hello everyone, my name is Cindy Lopez and I will be presenting topic number 2 for our individual presentation number 3 in cell biology, transition temperatures or TM. So what is a transition temperature? It is defined as the temperature at which a cell membrane becomes fluid when warmed from a gel-like state. A great example of this is the melting of butter from a solid to a more liquid form when warmed. The act of transitioning is called phase transition. Here are listed some of the requirements that a membrane must go through in order to function properly and not function properly. To function properly, the cell membrane must be in its fluid state. This means that the temperature has to be above the Tm value. And for the membrane to not function properly, the membrane is in its solid state. This means that the temperature is below its Tm value. Usually in the solid state, the function of the membrane is impaired, such as cell-to-cell communication, detection and transmission of signals, and transports of solutes. I've also provided an image taken from our textbook that gives you a better idea of what TM is, and it basically demonstrates that the peak of this line indicates the point where a membrane goes from a solid-like state to a more fluid-like state. This image is specifically taken from a TM measuring technique known as differential scanning calorimetry. For the next few slides, I will be explaining how different factors have an effect on TM and fluidity in membranes. To start off, how does the saturated and unsaturated lipid affect TM and fluidity of membranes? As you recall, saturated lipids do not have double bonds, while unsaturated lipids have one or more double bonds. It is stated that saturated fatty acids typically have a higher transition temperature, while unsaturated fatty acids have a lower transition temperature. This is the reason why saturated fats like butter and lard are in solid form at room temperature, and why unsaturated fats like oil are more liquid at room temperature. You can see from the image here how the unsaturated and saturated lipids are arranged. The saturated fatty acids are able to be packed well together, making them solid, while unsaturated fatty acids are not able to be arranged together properly because of the kink that forms from the double bonds, making them more likely to be fluid. This next slide goes hand in hand with the previous slide. How do short and long fatty acid chains affect the transition temperature and fluidity of membranes? Well, long fatty acid chains tend to be less fluid, meaning that they have a higher transition temperature. Short fatty acid chains tend to be more fluid, meaning that they have a lower transition temperature. This image that I included here depicts the amount of carbons affect fluidity in membranes. As you add more and more carbons, you increase the length of the fatty acid and you get less fluidity. How do sterols affect transition temperature and fluidity? Animal cells have large amounts of cholesterol. About 50% of the cells contain cholesterol. Cholesterol molecules are able to orient themselves in the membrane so that the hydroxyl groups closest to the polar head of the phospholipids are able to form hydrogen bonds. This leads to the cell membrane to become more rigid and less fluid at high temperatures. But cholesterols can also prevent membranes from gelling up at cooler temperatures. Because of this, cholesterol is known as a fluidity buffer, decreasing the membrane fluidity at temperatures above transition temperatures and increasing membrane fluidity at temperatures below the transition temperatures. This image describes how cholesterol orients itself in the plasma membranes of an animal cell. Finally, I'm going to describe the different strategies that different organisms use to maintain fluidity. Some organisms can regulate fluidity by changing the lipid compositions of their membranes. This is mostly seen in poikilotherms, or organisms that cannot regulate their internal temperature. Such examples of poikilotherms are bacteria, fungi, protozoa, algae, plants, and cold-blooded animals. The strategy of regulating fluidity is known as homeoviscous adaptation. Some examples of homeoviscous adaptation is when bacterial species at low temperatures can trigger the synthesis of desaturase enzymes. This enzyme introduces double bonds to the fatty acid chains to help lower the transition temperature of bacterial membranes, thus ensuring fluidity at low temperatures. Another example of homeoviscous adaptation is in cold-blooded animals like reptiles, where they can increase the amount of cholesterol in their membranes, thus decreasing the chance of their membranes going to a gel-like state. Thank you so much for listening to my presentation, and I'll see you guys on the next one.